Thank you for guys to stick with us until the end. I was I know I was told that there was various requests to understand how to model planar cell polarity using dynamic compartments in CompuCell. That's basically an extension of the compartments lecture that you have seen yesterday. So the way this so I have to just give a caution that I just put up this presentation from yesterday to today, so it's not completely done. I think it could have been better, but I ask you for your patience and and bear with me as we go along. Okay. So again, this meeting is being recorded and live streamed to YouTube. If you have any questions or problems or issues, please reach us in the chat, and someone can help you either through the chat, or you can go to a breakout room to work anything in more detail. All right. So we have seen yesterday that the cells are not a homogeneous bag of fluid. Okay, they have a structure, and sometimes you want to model the structure in CompuCell by, instead of modeling a cell as a single object, you model your cell as a collection of objects, which we usually call compartments. And these compartments can either reflect a physical structure in the cell, like the membrane or the nucleus, or can reflect some kind of more abstract region of the cell, as the apical and basal compartment of a cell, or the laminipodia and the cytoplasm, or the rest of the cell that's non-laminipodia, in the case of a cell that's moving. Okay, so this is how we did that's the one simulation that we did. Here is just a movie of a three-dimensional epithelia, where the cells are actually doing something called convergence thickening. They are getting longer and taller. Okay, there are other tissues around, but that you do not see in this video. And one thing that we did was today actually was to set up a epithelial, and we are modeling this epithelial sheet with using of compartments. Okay, so in all those compartments, the cells are still moving. They can still move around, but the compartments themselves, they are not dynamic, in the sense that once I place my apical side on the top, and I blue is basal compartment on the bottom, they stay there forever, right? So if we were just to pick up a cell in that epithelium back here and flip it, okay, the cell will be very unhappy, and will not be able to repolarize. Okay, the cell will have to go a full rotation to get back into shape. While I'm not sure that if this experiment can be done in reality, but what you expect is that the cell will just kind of, instead of rotating, you will just kind of redistribute its internal molecules and surface molecules to adjust to the environment. Okay, so this cannot be done readily if you just use the compartments as you learned on the previous lectures. Okay, that requires use of dynamic compartments. Okay, so, and this dynamic compartment is something that we have already been using to some extent. For example, in this simulation here that I just showed the screenshot at the beginning yesterday, is actually something you see. The cells are polarized. You can see there's a center apical side, the basal side outside. There's a non-visualized lateral side here on the sides, and the cells are staying there. But you notice that the cells are a little bit pixelated, is because I set up these cells in such a way that I'm not forcing by hand those compartments to be either here or there, or to the side. I'm just making those compartments actually to be preferentially mixed inside the cell, and let the adhesion contacts guide where each one of those compartments should assemble. So it's a self-organizing. I model the cell compartment as a self-organizing system, if that makes sense. So that may be better appreciated if I maybe play this next movie. You can see this is a movie that actually you have a cell that's growing and forming a cyst, and you can see that the resolution is not very good, but you see that all the compartments are mixed here inside the cell. And the first thing that the cell does is just kind of rearrange its compartments in response to the to the fact that it's inside some kind of buffer or medium or liquid, okay? And then as the cell grows and divides, you can see that those auto compartments start to sort out towards the correct positions. There is a mechanism to create a lumen inside the cells and create a cyst. And you can see that it's self-organized. I don't have to do anything. Once I set up these dynamic compartments, the system self-organize and go to the correct place. So if I just play again, you can also notice that when I divide the cell, uh, here maybe you can see that some of those snapshots, not here, I didn't quite capture. You have to be a little bit lucky with the let me see if I can. Okay, it was kind of hard to catch it, but when the cell divides, what I do, I mix all the compartments inside the cell and let them resort back then. There's nothing inside the lumen. The lumen is just this dark green cell in the middle. Well, the lumen is just an empty space filled with some kind of liquid. How you overcome the fact that sometimes the compartmental cell division um, break clusters apart? Okay, that is by a careful choice of the energy that used for the contact and the contact internal. Uh, well, in this case, because I'm mixing all the cells before I do the division, I don't have this issue, okay? Because it always replicates and resorts to the correct place. And here is a case where this actually grows before dividing. So the issue of just dividing a cell arbitrarily does not happen because the cell grows, all the compartments grow in accordance, and so when I do the division, everything gets I have enough material to do the division. Okay, so I don't see this problem of the compartment, some compartments missing by deep end sorrow. Yeah, more uh, right in the simulation, it was a little bit of that. Yes. Okay. So another example of use compartmental cells is in this movie that you may have already seen, but much reduced. So this one is a case where you have a mesenchymal cells that undergo epithelial transition, mesenchymal to epithelial transition. So a mesenchyme cell has no structure, right? Yeah, I just modeled as those initial blue cells, and then suddenly they undergo epithelialization. So here in this model, epithelialization happens suddenly, in a very abrupt way, okay? And but when you go to epithelial cells, I have to place a compartment somewhere, okay? And that is a problem here because uh, if I start the compartments to place in a specific place, I may be adding a bias to my whole simulation, okay? And then, and one of the objectives of this particular simulation in particular was to show that you don't need any kind of external bias or cue to to make this epithelialization and the formation of these epithelial balls. Okay. Uh, so here, what I did, you are missing the exact frame where this happens. What I did is I create those new compartments and I run them distributed inside the cells and let the cells interaction with the medium, with the next cell, and with the small luminal fluid that's already pre-exists within the cells dictate where those compartments should sort out. And then the cells also have a internal elongation mechanism, okay? And then this completely gets rid of any bias except for the existence of external boundary, and the system self-organize, and you start forming these epithelial balls. Okay. And the other thing that I can do with this development compartments is sometimes the cell is very unhappy, and what it does is completely depolarize, it loses its links, repolarize again, and then resort out and try again to go to one cell, to one epithelial ball, and can actually go jump to the next epithelial ball. Okay. Can you share this CompuCell3D file epithelial ball formation? Uh, Yes, I have no problem sharing it, but I don't have it in the new syntax code for CompuCell that was done in an old syntax. And so I have to first translate the model and make sure that's very legible to people, because when I left the group, some people, it was everybody complained that my code was not very uh, friendly. But in my defense, I have to write this simulation in a month, otherwise I will not be in the paper. So I did in a very rushed way this. Yes, so but that's something that we're gonna take a while to do. Okay, so the plan for today is to uh, tell you what's going on, what is behind this model, where you actually can do models plus polarity. And on this one, you are on this simulation where you can see that the tissue is reorganizing and making a polarization in the whole tissue is uh, is using this dynamic compartment. So what's going on is that here you have a periodic simulation with periodic boundary conditions. So if you go to the left here, you reappear here on the right. If you go to the top, you reappear on the bottom. Okay, and you can barely see the cell outlines because all the cell compartments are placed randomly inside the cell. Uh, so what happens is that those different colors here means different types of molecules that you have in your cell. The white is gonna be just kind of the cytoplasm. Blue will be the other types of molecules that like to stick to the neighboring cell, and then red will be all the collection of molecules like Van Gogh and Prickle that polarize to the proximal side of the cell, and the green will be the collection of molecules like Frizzle, the Shelf, and Diego that actually polarize to the opposite side of cell. And the way, and this is something that happens a lot in development biology, is a way to polarize a pre-existing epithelia. So there's two break of symmetries that can happen in the cell, right? Well, there are many types of symmetry breaking that can happen when the cell moves, is breaking symmetry. When the cell epithelializes, its breaking is apical to basal symmetry, right? So epithelial cell has a broken symmetry because it has an apical side on the top and a basal side on the bottom. And with that, you actually can form an epithelia. On top of this apical basal polarization, you can also have this in-plane polarization that happens mainly on the apical side of the cells. Okay, and because this polarization happens on the plane of the epithelia, that's why you call it PCP, planar cell polarity. Okay, so this planar cell polarity is just making your pre-existing epithelium to now to be oriented on the plane of the epithelium in a particular direction, and that can be used to set the properties of the cell, to drive, for example, cell intercalation events, or to just orient the direction at which little feathers will be oriented in your epithelium. So your cells can actually make a little bit of a feather out of it, and you want it to be usually slanted towards one direction. So that's what's case is happening on the outside epithelium of the wing, of the fly wing. Okay, so the flywing, when it develops, it has an outside epithelium, right? And outside epithelium is actually polarized. There's this kind of planar polarity mechanism going on there, and that orients the these little feathers that exist on the wing of the fly. Okay, these little spikes that come out. So this is a um, uh, image of the epithelium when you actually can see those little feathers getting out. Okay, and here you can see the polarization of these molecules to be oriented either towards the proximal side of the cell, or in other set of molecules be oriented to the other side, to the distal side of cell. How exactly you achieve this polarization? It used to be well, as far as I know, it's still something that's being studied. It can either be a self-organized event, like you see in this simulation that I have here, where uh, uh, those green molecules that are on the membrane like to make a heterotypic connections with those green molecules that are on the other side of the cell. And if all the cells are trying to do that, you may have a situation where you can actually self-organize the whole tissue to be oriented in a certain way. So this is actually what's happening on this simulation, okay, and where everything has no bias. I'm using a pseudo infinite epithelia because I have boundary conditions, and then the system kind of those molecules start to self-organize, those compartments start to self-organize, and in the end simulation you have a very well-oriented tissue where if I plot a vector connecting the right red compartment towards the green compartment, all those vectors are pointing the same direction. Another way that you can achieve the same thing is by maybe is self-organization, but there is an initial bias as of some kind of wall on one side that actually bias all force orientation on those cells that may be spread to all the other cells. Or maybe you can also have this wall can be actually activating those cells in a kind of cascade, and then so after a time the cells become activated, the next cell row becomes activated, and this would be a very robust way to actually polarize the whole tissue towards the same direction. Then you can see here the simulation, and on the right just a way to visualize this vector field of the cells. Okay, is this making sense so far? um, okay, so before we get into how we can model this in the planar polarity, we have to do some. My plan today is to do some exercises modeling a single cell with dynamic compartments, then two cells with dynamic compartments, and then show the one version of the planar polarity code where you have this kind of mechanism where the cells self-organize, but there is a boundary to the on one side that helps it orient the first row of cells. Okay, so let me check my watch. So the I have this picture here of those the use of dynamic compartments that was extracted from my PhD thesis. Okay, and our objective for the first two exercises is to recreate all those uh, pictures here. Okay, all those snapshots, all those panels. The first exercise will be dealing with a single cell, and you want to first recreate this one, and recreate then this one here. Okay. Uh, so for this first exercise, let's already start with a pre-existing model. It's you can be found on this Google Drive link. I'll paste that on the chat so you can download and upload to your NanoHub or use on your local machine. So let me just grab the link here real quick, and I'll paste in the chat for you. And is the link for the zip file. Okay, the link is on the chat. Uh, so I'll give a minute or two for you guys to download the basic simulation and try it and open up in your own machine, be your local desktop or laptop or your in your Nano Hub. So did anyone who had already got the simulation and have it open on your tweeted? Please let
Is
me
it
know.
4 cells in a cluster?
Yes, yes. Well, it's a single cell with 4 compartments, right?
Okay.
So when you get to this cells with multiple compartments, then you have to be careful with what you call a cell or not. Okay. So once you have compartments, I like to call uh, compartments and the cluster. Or, and the whole CompuCell3D was built assuming that each cell will be modeled with a single type of domain, right? Um, but once you start to make those more elaborate models, then actually you're using 2, 3, 4 domains, right? So I like to call them domains to be more generic, and only call a cell can be made of a single domain, or a cell may be a cluster of multiple domains, and those domains will be the compartments of your cell. Okay, I see that at least 9 people were able to download it. Okay, so if you have already downloaded, you can just open that on Tweedit, and I can go on with what's going on that code. Okay, so it's made of 4 compartments, right? Uh, and basically it starts with and all the contact energies are the same. Okay, so we have all the contact energies set up to 10. Okay, and the internal contact energies I place on the top all the internal contact energies between same compartment types that they actually have no effect on simulation because you don't have more than one type of compartment inside the cell, so they are all set to 0. And we have already agreed before that if you're using external and internal contact energies, the internal contact energies have to be lower than the external contact energy. So I made this non-working model where all the external contact energies are 10 and now the internal contact energies that matter, right, are set to a lower value, in this case 2. Okay. Uh, so in other words, if you just run this simulation nothing really interesting will happen. Okay. The other thing is that this just set up the energies, and you may have seen that in the uniform initializer, I'm just creating a single cell, which is a cell of 24 by 24. It's a very big cell. Okay, so when you use this kind of compartments, you have to be using a bigger cell than you normally would do. So I actually create those. So I start with a single cell of type C. Okay. By the way, the cell types are C, which will be stand for cytoplasm, and the other cell types or compartment types will be B, R, G, which is going to be the blue, the red, and the green. Okay. What the blue, the red, and the green exactly are is up to you to decide. Okay. Um, but it will be already useful if you run the simulation in the player, you already change the colors of your cells to be white, or sorry, yellow for the C, which stands for cytoplasm, and blue for the B, red for the R, and green for the G. Okay, so the place where I actually add all those different compartments inside the cell is on the steppable file, and this is basically what's going on there. Okay, so I do a loop over all the cells of type C, which is the cytoplasm, and there's only one cell in this initial simulation. Okay, and what I do once I get that cell is I first use this command to create a blue, a red, and a green cell. Okay, so this will just create those cells, but the cells they do exist in the simulation now, but they do not exist in the grid. Okay, so uh, so the next thing I do is I make sure that the blue, the red, and the green compartments now belong to the same compartments, the C cell. So I reassign the B, the R, and G to have the same cluster ID as this cell here. That this is what those three lines are doing. And now here on this set of code, I'm going over all the pixels that made up this initial cell, and I'm assigning randomly those pixels to be either to belong to the blue, to the red, or the green cell. Okay, so here you can see, maybe this is the first time you're seeing it. If you took pre-on parallel session, you have already seen that before. But basically, I'm getting a list of all the pixels that belong to this cell. Okay, and then I'm going over all those pixels. Okay, on the pixel list. So, and because I choose the simulation, I'm just retrieving the X and the Y coordinates of those pixels. And once I have the X and Y, what I'm doing here, I just create a random number. And this random number is actually be chosen from this list. It's going to be either 1, 2, 3, 4, 5, or 6. I'm just pick up one of those values at random. And the following lines here is assign the blue, the red, or the green based on whatever you have. This picket random number is either 1, 2, or 3. Okay. Uh, if my random number is 4, 5, or 6, I don't do anything, which means that half of my cell after this pixelation will be of types blue, red, and green, and the other half will remain as the type cytoplasm. I want this compartment to be bigger in my cell, okay, and the other ones to actually will be the molecules that actually go around the cell, that will go to the surface. Okay, so this is just a way to initialize the cell. So I drawing those compartments at random positions. So it cannot be more random than this. And after I do that, I'm just setting up the target volumes. Okay, so the target volumes of the original cell, the blue, the red, and the green compartment will be whatever volume they end up being after this uh, pixelation algorithm here, and then I set their lambda volumes to be equal to 2. Okay, is this making sense so far? Is it clear what's going on? If you guys have any questions or comments, please feel free to type in the chat, or just unmute yourself and ask the question. You can also raise your hand over Zoom and ask the question. Okay, if everything is clear, you can. So, okay. Yes, Sharon. So the pixels from the cell C, which the one that we started with, are just being reassigned to R, B, and G randomly. Okay, exactly that. Any more questions? So if not, you can just go ahead and play the simulation, right? Um, and if you play the simulation, you may get something that looks like a beach ball here. Uh, you get something that looks like this. Okay. Again, if anyone needs help or the simulation crashed on your computer, it shouldn't have done that. But if it did, let us know and someone can help you on the breakout room or just through message in the chat. Okay. So you know that all the internal contact energies and the external contact energies are the same, right? So you guess that the cells should preferentially mix, but that actually does not happen because the pixel copy events, right, are happen they happen based on the pre-existing pixel. So because the red pixel can only appear next to a previous red pixel, you have still some kind of spatial correlation between those domains. So they are not, even though all the internal energies are the same, they end up kind of sorting out into clusters like this, as you see on the screen, and hopefully as you see also in your computer. So let me just run a poll to see how your guys are doing with the exercise. So So were you able to download the questions? Were you able to download the code, look at the code, and run the code? And if you run the code, did you get something that looks like this? Okay, just let me know real quick.
Julio, you said something that I don't know if I agree too much. You said that we should expect the cells to mix, but we still have positive contact internal energies.
Yeah, that's true. That's true. Yeah, that's a good point. I disagree with myself and agree to you because they still have positive internal contact energy. They should sort out. Um, yes, I agree with you. So it seems that most of you got to this point. So let's proceed, and then I'll give you the first task of the module. Change the content energies to actually drive the mixing of the compartments, as you see here on the right. Okay, I'll give you a few seconds to try to do that, and let me know if it worked or did not work. If it worked, what value of internal energies you used. If you tried and did not work, also tell me it did not work and I used this value. So who got the cells to mix like this? Okay. because they are part of the same compartment. You should choose change those values here in the internal contact energy. You don't have two cells, so the only thing that's being used on the contact uh, plugin are those values here that set up the interaction of those compartments with the medium. All the rest here are being ignored. They are those compartments are interacting through those energies here, okay, the internal ones. 200. Well, you said the contact energy to be very, very, very high. Actually, that will drive separation of uh, uh, those cells instead of mixing them. Okay. Okay. So one thing that you may have done is the following. You may have just tried to make those additional energies to be zero. Okay. Because if they're zero, they don't care if they actually are next to the pixel of their own compartment or to the pixel to a different compartment, right? However, just setting the internal energy to zero actually does not quite get there because exact what I said before. Uh, the pixel copies can only happen from an existing pixel of that same type. So there is a history. So there's always have this little correlation between where the a blue pixel is and another blue pixel is. They are still spatially correlated because of the way you update the pixels. Okay, so to actually drive the whole system to mix, you have to make it actually in the cellular pulse GGH methodology. You have to actually drive that, push that a little bit further, and you can do that by actually making those contact energies negative. Okay, when you make those contact energies negative, then you are actually driving the mixing of all those compartments inside the cell. So for example, if you use the J to the internal contact energy to be -5, then you actually can get something that looks like on the picture on the right here. So that's a way to actually, when you're using those dynamic compartments, you want them to make them negative, so you can actually mix all of them together. What is negative contact energy? Does it have an actual meaning? Well, the best meaning that I can extract from here is that it's is a way to say that th- those objects that you're modeling, uh, they are just diffusing inside the cell, and it's more of a hack. Is a way to make sure that they're all uh, uh, mixed inside your cluster, than to then really have a biological or even physical meaning. Okay, is a indirect way to model compartment diffusion inside a cell. But when you're talking about not using this without the context of trying to mix the compartments, your simulation cells can actually have negative contact energies. Okay, and yeah, James is saying this right now, and actually there are very good reasons for to use negative contact energies in your CompuCell simulation. Um, I don't have time to explain that, but you uh, you can use negative contact energies in CompuCell simulations. When you do that, you also have to turn on the surface constraint plugin to prevent the cell from fragmenting. Okay, but the advantage of using negative contact energies is that the cells that stick together they preferentially stick together. They're not necessarily exchanging pixel copies as that often. Okay, so right now if you use positive contact energies, a high contact energy means those two cells separate and they become their boundaries become very stiff, while the cells that have very low contact energies close to zero, they stick to one another, but they also very dynamic in their boundaries. Okay, if you use negative contact energy, so you shift everything towards to be below zero, you kind of reverse those behaviors. The cells they stick together. They also don't have that many boundary movements between them. While the cells that are they don't like to stick together that much, they actually have a little bit more boundary movements between them, and that might be a little bit more realistic in some cases. Uh, there's a paper that discuss the advantages of using this technique. Actually, I just shared that paper with my new PhD student. It's a paper from 2003. Any more questions? Do you all got the cells to compartments to mix like you see on my screen? Okay, okay. So I do not know if I said anything wrong about negative energies. If I did, James, please come and correct me. Okay, so if you did this, then oh, that's fine. Okay, now let's give the second task. Let's make the yellow compartment, the cytoplasm, the C compartment to stay inside the cell. Okay, so which energies you have to change? Okay, to do this, think about it a little bit and then just try on your code. So the objectives go from here to here. Okay, I'll give you a minute to try. Sharon got it. Excellent. Maybe it's time for me to throw a poll about exercise completion. So something will pop up in your screen. Okay, Sharon, which energy values you change?
I changed the internal everything, all the internal engines energies with C to everything else to +5 instead of -5, and I also had to change the energy between medium and type C. I set it up to 50.
Excellent. That's exactly what I did. So you may think, okay, so that's thank you, Sharon, for sharing this. That's also something that I did. If you just change, so you may think, okay, if you want to have this cytoplasm to be inside, you want to have a very high contact energy with everything that's external, right? So you may change the cytoplasm and the medium to be have a very high value. Actually, want to be 40, for example. Okay, but if you just do that, you still have a lot of mixing inside your cluster, right? That might be something desirable or non-desirable. Okay, it really depends on your application. Uh, if you're modeling a cross section of the cell, you want to see this picture on the right. If you're modeling just a top view of your cell, the apical side, or maybe the basal side, or the lateral, maybe then you actually may want to have still quite a deal of mixing like here. Okay, but to get this to unmix this part here, you actually to have to reverse back those internal energies of the cytoplasm with the red, green, and blue to a positive value, exactly as you did. I think you even use the same number as I, 2, right? And when you do that, you actually can sort and get the cell to look like this. And now there is a very clear interpretation of what's happened. This is yellow, is the cytoplasm, and all these other colors uh, would be some transmembrane protein or a protein that sits right next to memory that may interact uh, with a different cell. Okay, can even be maybe the something that lives on the cortex of the cell. Okay, so so you get this. Yeah, okay, 5.2 is very close. Yes, I think if you even I think even 0 would work, uh, but we all got it. So you see that 9 people got it. So let's go for the next task, which is now let's make the red compartment to sort towards the outside. Okay, so that's the last task of this exercise. What change in the code we must do to get this effect now? Pedro said that zero energy works as well. Thank you, Pedro. Okay, so here the interpretation may be that, okay, maybe the medium is the ICM, and the red compartments represent integrins. So once the cell is surrounded by ECM, which is the black region, the integrins should start to distribute along the cell surface. Okay, so that will be one way that you can interpret this kind of self-organization of this layer. Then the green and the blue can be caderins on other types of transmembrane proteins that do not necessarily interact with the extracellular matrix. Okay. So let me relaunch the pool. This one actually should be a little bit easier than the previous one. Okay, someone needs help. Please reach out for help in the chat. You can privately message one of our instructors and they can help you on the breakout room. Okay. Okay, many people have already done the exercise. In my case, what I did to get this is just play with the external contact energies and set the energy between the red compartment and the medium to, instead of being 10, reduce to 2, and that already preferentially sorts the red compartment to be on the periphery of our cluster, because that likes to be more in contact with the medium than the other um, compartments like to. So the green and the red the green and the blue compartment, they have a contact energy of 10 with the medium. If you make now the red to have a lower contact energy than this, to be 2, but still positive, because you don't want that to fragment into the tissue, then you actually sort out the red compartment to the periphery of the cell. Does it make sense? Did everybody got this? Okay. If you did, then you can actually proceed to the next set of exercises, which will be to
Oh, wait.
yes, yes.
You set red. You reduce. Okay, I did the opposite. I increased it blue and green to 20.
That would be another way to do it. Very good that you bring that up, right? Uh, what really matters is the difference in energies, right? You can also do both, as Sharon did. You can either make the green and the blue to really dislike the medium, right? So that would be kind of a case where you are kind of induce the endocytosis of those molecules, of the transmembrane molecules, or the other way. Well, there's a different way to interpret the thing, of course. Uh, uh, well, at least for me, change the R to with the external contact energy of the red compartment with the medium from 10 to 2 was enough to drive uh, this phenotype. Yeah, but yes, just increasing the red and green with the medium to from 10 to 20, while keeping the red and medium to 10 should have a very similar effect. Okay, so let's proceed to the next exercise, which is to try to create now 2 cells and reproduce the image that you see here on the right panels. Okay, so here you already have some kind of interpretation what might be going on. Here are 2 cells with all the green compartments sourced towards their interface, so that can be some homotypic cadherin interaction. And here on the bottom you have both the integrins sorting out, and some kind of Frin and Fs interacting along the membrane of those 2 cells, and Frin and Fs are cadherin-like transmembrane adhesion proteins that actually prefer to interact with an opposite type of a molecule. So Frins interact with Fs, but not among Frins and Frins on the same side. Okay, so that's actually a way to actually get sorting out of tissues. Okay, at specific boundaries. So let's try to do those 2 on the right. So the first thing well, your first task is to create 2 cells side by side. You can use the exact simulation that you're using right now. You just have to go to line 92 and make sure that and change either the box minimum coordinates or the box maximum coordinates, so that you now have 2 cells side by side. For those of you that think that's too boring, you can even have 4 cells side by side, or have a row of cells. As long as it fits in your simulation space, you can do that. But at minimum for this exercise, you need at least 2 cells. Okay. So please change that. And if you change that and just run, you should have a initial condition with many cells. Notice that you don't have to worry about coding the pixelization of the second cell, because our pixelization code would work for all the C cells that you have at the beginning of our simulation. So if you have 2, that code that you have seen at the beginning on the start function of the stepple, we'll do on 1, and then you proceed and do again for the next cell. Okay, so if I can go back there, you see that this is done for one cell. If you have 2 of those cells, you do for 1, and then you do for the next one. Okay. So it's this code here is dependent on how many C cells you have at the beginning. Okay, so if you just have more than one cell and run simulation, you should get something that looks like this. Please let me know if you get to this point. I want to pull and relaunch. If anyone is doing more than 2 cells, also let me know. It'll be fun to see if anyone did it. You can have 3 cells next to one another, or 4 cells. Okay. So some people need a little bit more time. Any questions or comments? So if this is too easy for you, you can already go ahead and try to get those 2 pictures that I showed at the beginning. Okay. Or you can do that for more than 2 cells. You can do that for 4 cells. All right. So if anyone needs help, please reach out for help in the chat. Okay. So now, if you got 2 cells and they should not look very good, let's proceed to the second task, which change your energy so the C compartment stays inside the cell. It does not preferentially glue to the C compartment of the neighboring cell. Okay. So go from a picture like here on the left to a picture like here on the right. Okay, so did anyone achieve to keep the C on the middle? If you did, tell me what you have done.
Okay, can you just tell me the energies you used? Because I need to enter the breakout room.
Okay, I just then share the here. This is the solution that I found. I just made all the external energies, contact energies of the C compartment to be very high. So I set the C with C, C with B, C with R, and C with G to be 40. And once you do that, you prevent uh, the the yellow compartment from interacting with anything that belongs to other clusters. Okay. Does it make sense? Okay. So if you just, so that was done on the external contact plugin. Okay. There's no need for you to change anything in the internal. Oh, James just did the CC external energy and it seemed to have worked. Okay, if you make a very high value, it will work. Just the C to C. Yeah, but it's a little bit dangerous because you might still have a case where you can still have the case where this yellow is at the boundary directly interacting with those green and blue compartments there. Um, well, if it's just doing that work, then it work. But I'll suggest doing all of them because well, the set should not be interactive with anything that's external to the other. If you want it to keep in the middle, okay. But so it's interesting to know that just doing the top ones here actually worked. Okay, thanks for sharing that. Okay, so if this is done, then we can proceed to the next one, which, okay, now that you have a kind of well-behaved two-cell compartments model, try to recreate with what you see here on the B and on the D panels of that figure. Okay, so that will be the last task before we move to the PCP model.
Julio, I think something went wrong with the screen share here. The slides are not, I'm only seeing half the slide. There we go.
Okay.
Is that better, Norma?
Okay, sorry for that. Oh, there's an update here from Sharon that you actually need those four to be. Okay, let me. You actually need all those four energies to be high because you saw exactly what I already described. So thanks, Sharon. So yeah, the problem is that this boundary between those being white is kind of really hard to see. If you make the boundary to be black, you should be able to see the interface between the two cells more easily. Unfortunately, the player cannot. There is no way in the player to adjust the thickness of those cell borders. It's always one. It is always the small resolution you can have in your screen. but if the CompuCell3D developers are listening to me right now, please add the border thickness option uh, to the player. That would be very useful. No. Request denied. Okay. Okay, so let me see how people are doing the exercise. So I lost the pull feature is not working anymore for me. Uh, this slide, Sharon, or the slide of the exercise?
Sorry, you had already done it actually. The one from the exit. Yeah, that was the one.
Okay, okay,
Yeah.
no problem.
Your screen share is messed up again, Julio.
Okay, let me just fix that. Okay. Did you fix?
Yeah, it looks better now.
Okay. Okay, so I think that a lot of people have already done it. So I just share now the what I use to reproduce those two cases. So on the left one, what I did is I just reduced the external contact energy between the. Okay, I think I pasted this code not quite in the right way. The two things on the bottom actually should be reversed. Okay. Okay, I'm sorry for that. I mixed up. Uh, okay. Let me just stop sharing a little bit, so I don't want to have this wrong on your screen. Okay. Okay, now is the correct solution for the exercise. Sorry for that. So for the B1, because I do not want the red compartment to sort towards the outside, I just set back that energy between red and the medium back to 10. Okay, and because I want the two green cells compartments to actually sort towards each other at the cell boundary interface, I set their contact energies to be 0, and then they preferentially go there, and then you have this interface of green to green compartments. Okay, then for the next one, what I did, because I want the red compartments to be towards the outside, I set make sure that energy it's still 2 with the medium, so a low energy that makes the red to go towards the medium, and then at the interface between the cells, I'll make the contact energy between the blue and the green compartments to be 0. Okay, and the green is set back to 10, and with this I induce the sorting of the green compartment in one cell to be touching the blue compartment on the other cell. Okay, so by using these compartments that preferentially mix inside the cell and choose the correct contact energies, you can actually induce the sorting of these compartments to the correct place based on their environment. Okay, so if you do a simulation now with 4 cells, for example, this case, you should see more or less the same pattern. Blue secreting towards the green, and the green secreting towards the blue. Okay, because you have a finite number of pixels away for each compartment, you may have a case where one side of the cell, or some sides of the cells, all the green compartments go there, and all the blue compartments go to the other side of the cell. Okay, but you can see that the interaction, what made those compartments inside a cell to sort was not something internal to the cell. It was the interaction of the cell, the cluster, with its environment. So the environment was providing the cue. Okay, and in the case where you have 2 cells, or if you just fill up this the whole simulation with cells, okay, you have a situation where at first you do have no symmetry. The compartments are just randomly distributed, and the self interaction between the cell compartments with the external compartments will kind of set up some kind of pattern that that emerge out of your system. Okay, is this making any sense? So that is one way that you can use those dynamic compartments. Okay, so is a technique to actually have those things to is another way to create emergence, self-organization in using the cellular pots model. Okay, where you're modeling the cell, and in some abstract way, its internal components. So I like to call this the subcellular pots model. Okay. So any questions or comments? If no, let's proceed, and let's try to make a simulation of planar cell polarity. Let's see how. Don't have much time left, but here the plan is to actually I want to share a code that does that with you. I was pasting that in the folder right now, and let me just step back from here for a few seconds to get the link back, and I'm gonna place the link in the chat. So I will give a code that does something that's similar to what you have on the screen, and the plan here is not really to do any exercise. I will just go over the code, tell you what's going on, and then it will be up to you to do your own type of exercise on top of it. Okay, so while I do this, are there any questions that I can answer for you. Okay, so the link to download the file is in the chat right now. Okay, please take a time to click on the link and download it and upload to your NanoHub or your desktop machine and try to open that on Tweedit. Okay. So do you see the link in the chat? So I just launched a poll to see how many of you got the file in the computer. So if you got the file in the computer, you can already open it and tweet it. You can even start running it to see how it looks like. And I'll then share my desktop and go over what's going on in the code and in the simulation. Okay. Okay, so I'll stop sharing my screen and move on to go to my desktop to okay. So for those of you that got the file, did it work? I hope it did. So let me know. Yes, okay. Thank you for answering. Okay, I see that already a good number of people got the file to work. If you got it to work, please answer in the posts for me to get an idea how many have the file installed on their computers, okay, and running. So I'll now do a share my my desktop. What I want to see is, wait, where is it? Here, this is what I want to show you guys. So are you seeing my screen? Okay. So let me go directly to the simulation. Okay. If I just run simulation and I just click on the step function, what you should see is is a bunch of cells that are pixelated, okay, with some kind of big green domain on the bottom, okay, and here you also see a graph, okay. You can see that this graph has X and Y, just like this window here. And what is happening here is that I have some kind of boundary here, a green boundary, and then there's a black outside boundary. I just have 4 row cells. I have my cells have a yellow domain that stands for the cytoplasm, right? No domain that does not interact. Red domain that's called the proximal domain. A green domain that I will be calling the distal domain. So this has an axis. So this is the proximal. This is distal, okay, and the boundary is on the proximal side. And then you also have a blue domain that just stands for lateral, okay. And then when I run simulation, those pixels start to self-organize. And what I'm doing here on the left in this graphics session, I'm using a visualization field, which I'm not sure if someone actually covered that in details, but you can actually create visualization fields to either color your cells with different colors based on properties, or creating vector fields like you see here. And here in this vector field, I'm just create a quantity, a vector, where I basically get the distance from, or the direction from the red compartment to the green compartment, create a vector of that, and then I just visualize here on the visualization field.
Can you show the setup code for that? Because that's not something we showed elsewhere.
Yes, I'll be happy to do. So this is if you play the simulation, this is what's happening, and you can see it all those vectors here aligning, showing that your cells compartments are actually sorting out to be all bit from this side to this side. And what is going on is exactly what you have been playing before. Is you're using the dynamic compartments of the cells. So let me now pause here and let's go to the code, see what's going on. Okay. So let me close this project here. So let me go to the XML file. You just set the dimensions of the properties. So I have 4 different cell types. A core cell, which is going to be the yellow part that you see, the proximal compartment, the distal compartment, and the lateral compartment. Okay. You have volume, center of mass, pixel tracker. Here will be the contact energies that I use to ensure sorting. Okay, and you can see the proximal and distal have a very low energy compared to the rest, so they like to be, the proximal side of one cell likes to be next to the distal compartment of another cell. And then the internal compartments, I've used the same trick as before, negative to induce mixing. In this case, I want the proximal and distal actually be not interact, so I actually set up a positive internal energy between those two, so they actually go to opposite sides of the cell. And other than that, I just have the initial, the uniform initializer here to draw the cells. And I'm doing the same trick as before. I'm just drawing the core cell and using on the XML file the Python steppable and just pixelating those cells. So let's now go to our Python file. I have 2 classes, one for the visualization and the other where I set up the initial conditions. Okay. So in the visualization, the initial conditions, I'm doing basically something that's very similar to what I had before. I'm going over all my core cells that I had at the beginning, and then I creating those new compartments, giving them a fixed set of target volumes, okay, and lambda volumes. I group them into clusters as I'm doing here. This is something else that will be used when I draw that big green compartment at the bottom. And here I'm just doing the same trick as I was doing before. I'm going over all the pixels that made of the cell and randomly assigning proximal, distal, or lateral pixels to it. Okay, so it's exactly the same thing as before. And at the very end, I'm just drawing a big distal compartment, the green compartment at the bottom of the simulation, to actually have a some kind of boundary to induce a break of symmetry that will induce all the cells to actually be oriented in a certain way. And if you watch the move closely of those visualization factor, you see that the first row becomes oriented, then the second row, and the third row, there is a spread of information as it happens in the simulation. I guess many of you want to see how actually you get those vector field. Okay, this is done, implemented here on this section. So the way that you get here is you can right click on the steppable files, add steppable, and in your steppable name you can actually make that a visualization field, and it can be a vector where you can, for each point in space, you can assign a kind of vector, and the vector will just pop up there. Or it can be a scalar. You can color a cell. You can color different pixels with different values, and that will be show up in the CompuCell simulation. It can be a scalar cell level where you color each cell or each compartment with some kind of value, and that will be shown as a color map in our visualization field. Or it can be something that I use here, the vector at the cell level, okay, where I associating each cell with some kind of vector, and the player visualize those vectors. So each cell will have a vector, and the player visualize it. Okay, so that's how you get to this. I'll not do this now. So here I select the cell level vector. Okay, so you get this, and the first thing you have to do, you have to create a kind of object where you're going to be create vector field at the cell level, Python, and this is going to be the name of my vector field, and that's the name that appears here in the drop-down menu. By definition, you have a cell field, and as one way, things that you can visualize. Once you create these other visualization fields, this menu here actually be pop up with all the visualization fields that you created. Okay, so here if I select PCP vector, I'm now seeing the vectors that I'm creating. So let me show what's going on here. First, I'm waiting. Okay, there's a question. Yes, they'll be shared. Okay, I did not upload them yet. Sorry for that. I'll be loaded as soon as they are done here. Okay, I promise that I'll do that. Yes. So here, after a Monte Carlo step of 100, I updating the field. So here I'm calling line 65, where I have to clear the field for any vectors that I have drawn before. Otherwise, they keep just accumulating there. So here this comes to I'm calling this field object, and I'm using the command .clear to clean it. Okay, so that will be empty. And then what I'm doing, I'm going over all the cells that score are of a core type. So let me just show here. So what I'm basically doing, I'm going over all those yellow compartments, okay? And then I'm inquiring where the red compartment is and where the green compartment is, or where the proximal compartment and where the distal compartment are. So I get the list of cell clusters. Well, this actually should be called list of compartments of the cell, okay? And then I go over all the compartments that belong to this one here, and if they are a compartment type proximal, I store it, if it's a compartment of type distal or the green compartment, I also store it, okay? And once I completed this loop, I got a hold of the P compartment and get a hold of the D compartment. And if I found both compartments, okay, so this just make sure that I do have a P and a D compartment in my cells. Then I ask the distance in vector form between the center of mass of one and the center of mass of the other, okay? I calculate the angle based on this set of distances, okay? Well, this seems a little bit, because I want all my vectors to be of the same size, I basically am normalizing this whole thing, right? Make sure that the vector will be of size 1. So that's why I take the angle and then I recreate a vector to be the cosine of this angle, sine of this angle, and 0. Okay, that if I do this for all the cells, even if some compartments are closer, some are further apart, the vector that I get in the end will be of size 1, of length 1. Okay, and then I populate my vector field by calling the cell field, give an argument as the cell, and I populate with this vector, which is going to be just a collection of x, y, and z values. And then what CompuCell does is automatically draw the vector in the visualization field. Okay. So is it clear what's going on here? Any questions? So basically, that's yes.
Can you change the arrow type?
No, unfortunately, we cannot change the arrow type. All the arrows are this kind of head arrow, but you can do some kind of there's always the option to do some kind of customization of your visualization fields. So if you just click here on this the toolbox, here you see your familiar cell color options. If you click on the field, you have here the PCP vector as one of the options that you can see, and you can change the color of the arrows to be pink, for example. Okay, I can hit OK. Now all my arrows are pink. Okay, but unfortunately, we don't have yet the preference to have arrows of different types. Okay. And also there is the option to scale the arrow length according to the size of the vector that you're plotting. Okay, so and what the CompuCell player also does is color the arrow based on its size as well. You can also do that. You can kind of overlay vector and cell field. If I let this option here, let me see what happens. Okay. Oh, actually it didn't do anything. Maybe I have to run a little bit in simulation. No, it didn't work. But there are ways to customize your visualization fields from the player. Okay. So this is any question about how the vector fields work or how the simulation itself works?
I have a question. So in my opinion, the next level would be obviously to build a 3D simulation. What are the main difficulties?
Okay, for the PCP, a 3D simulation makes no sense because this is something that happens on a plane. If the question is how to make a dynamic compartment in 3D, the question is just to make the appropriate choices for the contact energies. Okay. So the dynamic compartment, because it's using an internal function that's intrinsic to the cellular pot CGH algorithm, there's nothing really that you can do change. All the kinds of precautions that you have to take for going from 2 dimensions to 3 dimensional simulations will also apply to use this technique of dynamic compartments. And actually, that simulation that I showed at the beginning of the cysts forming is a simulation in 3 dimensions that use the dynamic cell compartments technique.
So it was trivial to generalize.
Yeah, it's kind of trivial. Okay.
Okay.
If you scale all the other constraints accordingly, is exact the same. Is exact the same thing that's going on.
Yeah, sometimes it is very challenging to generalize to 3 dimensions because, for example, in a cell crawling simulation, if you go to 3D, you have a substrate.
Yeah. Yes.
Now you have many more contact energies, and then now the size of the things matter more, etc., etc.
Okay.
So in this case, it wouldn't be trivial.
Yes. Okay. So actually, thanks very much for the question, Pedro. And actually, that's everything that I have planned for today. So if this module was a little bit since a little bit organized, is because I just prepared that on a rush from yesterday to today. So I apologize for if it seems a little bit less hectic. But I'm sharing here the code for the PCP. You can, now that you guys have the code, you can play with it. And one thing that you guys can do is maybe change the initial conditions to have the whole space filled with cells and see if all those cells will self-organize. Okay. Or you can add more cell layers and see if it takes longer for the whole tissue to become oriented. There are things that you can do, so it's up to you to do it. And please stick around for the Q&A sessions. I do ask you to please do not publish this model yet, because do not scoop me with my own model, but you are free to play and use on your projects. Okay.